about mitosis. Yay! Which is a process of eukaryotic cell division. Mitosis is super important. This is how almost all the cells in our body and basically every other animal and also plants bodies divide. So it's something that you'll definitely be tested on at the end of the year. So make sure to pay attention. So, like I said, mitosis is the process of cellular division. This includes us. And there is one little mnemonic that is going to make mitosis a lot easier for you, and that is PMAT. PMAT stands for prophase, metaphase, anaphase, and telophase. And if you remember PMAT, it'll make your life a lot easier, so go ahead and commit that to memory. Mitosis is also followed by the process of cytokinesis, and this is where the cell actually divides. So the big takeaway from mitosis is you start out with one cell and you end up with two identical daughter cells. So let's see how that happens. First, let's talk about the anatomy of a dividing cell. So just like all cells, this is kind of a cell before it's ready to divide. This is a cell just hanging out. We have our nucleus filled with chromatin. This is our nucleus. This is our chromatin. And if you remember, chromatin is uncondensed DNA. We usually draw it as something like spaghetti, but this is basically just when the DNA is like kind of wonky and it's not dressed up all nice, it's not organized really neatly. This is the DNA in the normal everyday functioning of a cell. We also have these little dudes here, which are centrioles and spindle fibers. And we are going to have more than one of these, and we're going to have two sets of these in our cell. So here we got these guys just hanging out. So if we look at these, we have our centriole. Oh, sorry. This is the centriole, and this is the spindle fiber. So during mitosis, the goal of, not spindle, mitosis, spindle fiber. So during mitosis, the goal is going to be to end up with two identical daughter cells. So we're going to be splitting the DNA, right? But the things that are going to make all of this possible are the centriole and spindle fibers. These are the workhorses of mitosis that are kind of making it all happen. So now we know what a dividing cell looks like. Let's start. Prophase. Prophase is going to be the very first step in PMAT. So P is for pro. And in prophase, a few things are going to be happening. Prophase, we're really gearing up for mitosis. We're getting everything ready so that we can divide. So one of the first things you're going to notice is that our centrioles are going to separate. And our spindles are going to form. So they're going to get a lot more robust than they were. So our centrioles and our spindle fibers that started out, oh no, I don't want to form that shape. They started out kind of near each other hanging out. I'm going to make them a little smaller so they don't take as much space. They are going to start migrating to opposite sides of the cell. So where in the last slide they were all kind of hanging out together, now they're starting to move apart a little bit. And then our spindles are going to start forming kind of in earnest. They were just sort of beginning, now they're getting a little longer and they're getting ready for the process of division. They've got a very important job to do and they are gearing up to do it. All right, the other big thing that you're going to notice is that before our nucleus looked like a normal nucleus. Now our nuclear envelope is actually going to start dissolving. Now, why would this happen? That's because if our DNA is contained in a nice solid nucleus, it's not going to be able to move, right? We're not going to be able to do all the acrobatics that we need to do with our nucleus if it is solid, because our DNA is going to be stuck inside. So we kind of got to get rid of the nucleus. Otherwise, all of our DNA is going to be stuck and we're not going to be able to do anything with it. So instead of having a nice solid nucleus at this point, you have a nucleus that's starting to break apart. It's got a bunch of holes in it, and it's eventually going to go away altogether. But in prophase, there's still a semblance of a nucleus. We're just not there yet. And finally, what used to be chromatin is going to condense into chromosomes. So we're going to draw, here's a little red chromosome. We'll do a few different colors because I think colors help a lot with tracking where things are in mitosis, and that's a big key. If you draw this at home to practice or study for the test, do it in color. It's really easy to get it very confused when you do it in black and white, and I find that practicing in color just makes it a lot better. So we have our little chromosomes. So the final big, very important thing that happens in prophase is that chromatin condenses into chromosomes. And chromosomes are these cute little X butterfly shaped guys. And these are what we're going to see for the rest of mitosis. All right, so this is prophase. Our centrioles are going to separate to opposite sides or move towards opposite sides. They're not quite there yet. Their spindles are going to form and get longer. The nuclear envelope is going to dissolve so that we're able to access our DNA, and our chromatin, the squiggly bits before, are going to get condensed in these nice, neat chromosomes. So moving on to metaphase. So metaphase is the M in PMAT, and meta means middle. So why is it called middle phase, basically? That's because all of our little guys from earlier, everything is going to go along to the middle plane of the cell. It's all of our chromosomes are going to line up along the equator. As you see, they're going to make a neat little line. It's going to be great, and it makes it really obvious why it's called, sorry, these are not the prettiest chromosomes, but they get the point across, why it's called metaphase. Meta for middle, and that is actually like a huge takeaway. So in metaphase, we're going to take our little centrioles and our spindle fibers. I'm going to make them smaller again because I don't want them to be giants. They have now fully migrated to opposite sides of the cells. Great. And then something happens here. Our spindle fibers, right, that are coming off of our centrioles, they are connecting to our chromosomes like this. And they are connecting, let's see here. The spindle fibers are what are doing the lining up in the middle. They're attached to the chromosomes, and they are making all of this happen. And they are attached to the chromosomes at a very specific point. And this point, this little dot in the middle, it's called the centromere. So let's kind of recap here what happens in metaphase. Our chromosomes line up in the middle, because middle means meta, or meta means middle. In the middle. And then our spindle fibers, our SF spindle fibers, are going to connect to the centromeres. And just to be clear on what a centromere is, here is a chromosome. At the center of the chromosome, you have this little bundle at the middle. This little bundle is the centromere. And that is metaphase. Metaphase is one of the easiest to recognize phases because everything is lined up all nice and pretty. So we've done prophase, we've done metaphase. Let's move on to anaphase. So anaphase is the A in PMAT. And anaphase is where our cell looks a little wonky. it's no longer a perfect sphere, and we're actually looking at more of an elongated cell at this point. It's starting to stretch out because it's going to be two cells eventually, and we're moving in that direction. So, once again, our little spindle fibers and centromeres are starting at opposite corners. Actually, let me go, let's see here. No, we'll just draw them. So something interesting is happening now with our chromosomes in anaphase. So they've actually started to pull apart, and they're no longer, so we used to have them connect in the middle, and we now are going to end up with two halves. And these two halves are each called sister chromatids. Sisters because they are, the same. They're the same little genetic information. They were once connected. Now they are not. Now they're just little sisters. And you can see how they used to be that one X formation that we had earlier. Great. All right. So these are our sister chromatids. Let me write that down for you. These are sister chromatids. They're no longer chromosomes, they are chromatids, right? Okay, because they've been pulled apart. And then our spindle fibers are still attached to each of the little centromeres. Boop, 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 boop. They're doing all the work, all our spindle fibers. We love them so much. Okay, so this is what anaphase looks like. Chromatids, you can think of them as they are little kids because they are cute, smaller versions of the chromosomes they originally were. So, wrapping it up, this is what happens in anaphase. Our sister chromatids are pulled apart. They are pulled apart by our spindle fibers at the centromere. And the cell is going to elongate. So that is another one of those big, if you're doing like a test where you have to identify which stage is this cell in, and they give you a picture. If it's starting to look a little like an oval, a little wonky, you can see back in metaphase, it's nice and circular. Now we've moved to a more of an oval. That's definitely going to be anaphase. And in anaphase, you're also going to have sister chromatids that got pulled apart, right? They're no longer one big chromosome. So moving on to the next phase, which is telophase. So telophase, because it looks like one of those old-fashioned telephones. I can't draw this. It's got like a dial and it like has, you know what I'm talking about? These? It'd be like ring ring. Yeah? No? Okay. Telophase is the final letter in PMAT. Okay, and you can see most obviously in telophase is that we're no longer even really looking at an oval shape. We now have this kind of like hourglass Coke bottle shape going on. So this is where we start to be like, okay, we kind of got almost two different cells going on again. Oh, we're almost there. So we have our centrioles and spindle fibers. Oh my gosh, I'm going to keep moving this apparently. We have our centrioles and spindle fibers, just like last time, just like all the times, except for now they're kind of starting to chill. So they're less involved than they have been because we're starting to wrap up mitosis actually. But we still have all of our chromatids, right? And they're still the same as they were in the last step. We have a red on each side, we have a blue on each side. And I would recommend that, like I said, every time you practice drawing mitosis, that you do it in colors because it makes it easier to keep track. You need to make sure that you are keeping numbers consistent when you do mitosis because numbers matter a lot. If you don't end up with the right number of chromosomes at the end, then your cell's gone through a pretty bad mutation and it might not survive. the next thing I'm drawing here is that the nuclear envelope starts to reform. Like I said, we're kind of starting to wrap up mitosis. So we're going to start putting back the things that we moved around to make mitosis possible. So we still have our chromatids, and these are, like we said before, they're our little kids. So telophase, to wrap it up, in this step, our chromatids are going to be on opposite sides, right? They're going to be gathering at the two different sides that are going to be two different cells at the end. And then our nuclear envelope is going to reform because we are getting ready to go back to being full cells now that aren't undergoing mitosis, right? Okay, so this is our telophase, the T, excuse me, in PMAT. This is the last step of mitosis. After this, mitosis is technically over. And you might be asking, wait a second, but we still only have one really wonky cell. We don't even have two cells yet. How can this be the end of mitosis? And that is because we still have one more step. It's just technically not part of mitosis, and that is cytokinesis. So cytokinesis happens after mitosis happens. So cytokinesis, let's look at the word cytokinesis. Cyto means cell, and kinesis means cut. So this is cellular cutting. And you can see that because we started out with one really wonky cell, and now we have two cells. So what do these two cells look like? Well, we know we still have these dudes. They're still hanging out. They just exist in the cell at this point. They're not doing, they help pull everything apart. And now here we are. We have them. We're happy with them. Great, love it. So let's copy these guys over here because we end up with identical cells. That's the point, right? Is we end up with two of the exact same thing. Our nuclear envelope is back. It's back and better than ever. So we have a nucleus and we have a nucleus. And actually we're just going to copy and paste because we want them to be identical. So, and in that nucleus, we are going to have all the DNA that we just split, right? And these are still looking as our sister chromatids. Amazing. And then remember, these guys are going to be identical. So it's just going to be, whoop. It's just going to be the exact same thing over here. And so with cytokinesis, at the end of cytokinesis, you have two identical cells. And that's very important. All things, if everything went well, they will be the exact same. And we actually call these daughter cells. That's the nomenclature, it's the standard. So during cytokinesis, here's what's actually happening. Our cytoplasm, if you remember, the cytoplasm is the goo inside of a cell. This is going to split in half. Okay? If you're wondering why cytokinesis isn't a part of mitosis, it's because mitosis is technically the duplication and division of the genetic material. Cytokinesis is the division of the cytoplasm. So it's just that the words kind of mean different things. Mitosis is always going to be followed by cytokinesis, but cytokinesis is not a part of mitosis technically. The other thing that's going to happen is that we end up with two daughter cells, right, that are oh my gosh, that are the same. And I know I've said that a lot, but that's really important because we are eventually going to learn about meiosis, where they are not the same. So we need to go ahead and master right now that in mitosis, you end up with something that is the same. All right, so now if we look back at our very original slide, we had this cell with our centrioles, spindle fibers, everything in it. This original cell, if this was the very first cell we started with, is going to be identical to the daughter cells. The DNA looks a little different because just in this picture, the DNA hasn't uncondensed back to chromatin yet, but all the DNA is the exact same. You just made copies of your original cell. Mitosis as the big picture. So this is the whole process of mitosis, right? So we're going to have our prophase, our metaphase, our anaphase, our telophase, and then our cytokinesis. And we start out with our one cell and we end up with our two daughter cells. And that is mitosis. Things a cell cycle, which I believe is review for you guys, but we are going to now connect it to what we've already learned about mitosis. So the cell cycle describes a cell's life cycle, right? How it grows and matures over time. So the first step of that is going to be G1, which stands for gap 1. And this is sort of just a growth phase of the cell. It's just kind of living its life, doing its thing. The next part of the cell cycle is going to be the S phase. And this is S for synthesis. And this is when DNA replication is going on. So this is where we go from having a normal amount of DNA to having twice the normal amount of DNA. Then we are going to move into another gap, and this is our G2 phase. And finally, we are going to have, and you'll notice it's really short actually, it's a very short amount of time, and this is our M phase. And this is where mitosis takes place, right? That's why it's called the M phase, because this is where mitosis actually exists. And you can see that if this was a pie chart, this is a really tiny slice of time in the total life cycle of the cell. What's going on in G1 and G2? We're prepping for mitosis some of this time. We're making sure we have all the supplies we need. We're growing, we're living, we're developing. The cell's not just sitting around during G1 and G2. And it's also important to note that the S phase happens before mitosis. If we didn't replicate our DNA, we wouldn't be able to split in mitosis because we would end up only having half as much DNA as we needed. So it's really important that this S phase happens prior to mitosis. Now there's one other word you need to know that pertains to the cell cycle, and that is that all of this, everything but mitosis, is interphase. So interphase, let me write it on here, we can actually read it, is G1, S, and G2. So it's everything but mitosis.